是我们还需要非常严谨的态度做每一件事情。所以这一点呢，所以我给大家就是说一下，就是从我们现在这个课开到现在，我请的每一个老师都是非常认真的在备课。我觉得至少比我在复旦上大学的课我要认真的多。为什么？因为我去到复旦大学，我们学生去上课，底下就没几个人，对吧？这没几个人，你说我备什么课？备半天，底下就没人听，对吧？所以这个可能效果还是不太一样。当然，大家可能也是冲着这个要学知识，我能够感觉到，就是包括Tom他们能感觉大家是冲着一种知识，就想来学这个东西。所以你想来学怎么都好了，你不想学你就来想，比如说上课就想拿个学分，到考试前问老师这个考什么东西，那这个东西哪有劲呢？对不对？所以这一点我觉得可能会。那今天 Tom 会讲 Excel， 这个其实是一个非常热的一个 topic。这个目前它的功能、它的机制，不清楚。而 Tom 是这方面的专家，他做这个东西做得非常多，因为他做 RN 这块嘛，所以这块做得非常多。所以我想今天的报告可能也会非常精彩。所以今天我哪儿都不去，我就听课了，对吧？所以这个我觉得可能还是比较重要的。然后另外从这件事情，今天下午我们的课本来安排Fred Tyson 他给我们做那个 就是 Epigen 整个的一个宏观的一个介绍。我们现在准备把它调在最后，就是说先可能是 proposal， 然后再是它的我想因为整个它相当于整个课一个 summary， 对吧？所以可能它会调在后面，所以大家可能知道一下这件事。我们1点半可能开始先是 proposal， 有6个人。我看了一下 proposal， 还是非常的。昨天，所以为什么要9号发过来？发来以后呢，Hillary 昨天就对我们要，就是说 Gingeras 的主编就是 Excel editor of Gingeras Research， 他一般情况下说实在话，你请他，别人请他去弄个什么，他可能就没空，所以他就像这个课，他也是真的把你们学生的幻灯拿过去。今天上午他也没过来，就在 working on this slide， 一个去看你的幻灯，想帮大家。所以我在想，就是说这次做 proposal 应该是比较 lucky， 你知道吧？昨天上午去，很多人想给他去输入什么东西，他都没时间听。但是这个他觉得是很认真的事情，所以一个 slide 帮大家看。所以我想今天下午我们这个讨论，我觉得国内可能也没有找一个很有名的期刊的主编来给你主持这种报告，我觉得可能还会比较少，所以我们也比较荣幸。而且他也不是就是说是人家搞算了，对吧？他把slide提前要去，他就看，还要去看怎么去弄。所以我觉得应该还是不错的，可能还是会能够帮到大家。这个今天下午的那个 proposal， 我估计中午的时候可能这个我们有非常详细的，就是我们当时原来说过，有50分是留给专家，另外5分是大家的，所以我们可能通过微信投票的方式，可能我们昨天做了测试，可能还是可以，就是说你们就完了以后，你们按投票，这可能是李伟具体介绍怎么去做，因为你们当时一按下去，因为很快我们那个就知道谁是票多少个，就等于说我们要不然的话会很慢，就是你们很快，你们弄完了后清完了报告，你们投票哗一投，我们马上就可以见到，就是那个统计马上就统计出来谁是多少票，然后可以统计，马上可能公布结果，就是我专家的票马上公布出来结果，就是最后的结果。然后我们后边课结束有一个颁奖仪式，应该还是比较好的，所以基本上目前是这样。那现在让我们热烈掌声欢迎 Tom 给我们今天上课。
Thanks to you for you taking time out to come and have this conversation together. I thought yesterday, we had the start of a conversation, and I hope we will continue that and maybe even increase it even more. Okay. All right. So today, I thought we would finish up one more topic which I didn't have time to speak about yesterday, and then convert into this discussion and presentation about the ability for cells to reach out beyond their boundaries and influence other cells, and what the mechanisms and roles are for that. So let me begin. So we talked yesterday largely about the roles of RNA primarily long non-coding RNAs, and to influence a variety of functions that are within the cells, and in some ways operate as if they were sort of the middle managers of information stored in the genome. And that middle management role, as we discussed yesterday, is a very powerful role because this is the only level where information is being passed to the genome, back to the genome, all right, through the mediation of RNA, as well as information from the genome being passed to the actuators. So this important role of non-coding RNAs is extended in such a fashion that it would be interesting to understand if there are other functions attributable to that sort of middle management capability. So one of the things that many of us who work in biology, in molecular biology, often use in our sort of determinations is the differential expression of a cell under different conditions, during development, during transformation, during growth, during replication. Those differentially expressed genes in that circumstance, it needs to be differentiated or separated from genes that are constantly being on and are part of the maintenance for the cell. So we call that group of cells that are important for the operation, the everyday operation of a cell, we call those genes housekeeping genes. But if any of you have ever looked into this concept of what is a housekeeping gene, right, in terms of what the elements what genes are expressed and at what levels are expression and why are they expressed, you'll look at literally 100 papers that mention these genes. And most of them have this very ill-defined sense of what a housekeeping gene is. Most of the time, the definition for that is that the level of expression is relatively constant from one condition to the other that's being compared. So for example, if you're looking at non-transformed cells versus transformed cells, right, the genes that have remained fairly constant in their expression level are often lumped together in this group that are called housekeeping genes. Right? Now, this serves a problem because when you look at the literature and ask, okay, tell me a list of genes which I can look at for any cell in any species that basically serves the function of housekeeping or maintenance of the function of the genes, you get very few sets of genes that are in agreement to everybody, all right, and many genes that look like they are specific to the cell type that is being studied, which is sort of not the definition of what people want to call a housekeeping gene. So we began looking at this issue in a sort of backdoor fashion. We didn't set out to actually ask the question of what housekeeping genes are, but the data that I will discuss with you now sort of led us to a principled way, a way in which reproducibly, irrespective of the experimental conditions or irrespective of the species that you're dealing with, will work and will give you what the housekeeping genes are. So let me tell you the story of how we got to this, this principled definition of housekeeping genes. We were doing an experiment. We've been working on the ENCODE human system for a long time. And then the mouse ENCODE system came up as part of the mouse ENCODE project. And so one of the things that we wanted to look at was the comparison of expression patterns and the regulation of that expression in mouse versus human. So we had a variety of cell lines in the human, about 18 cell lines, and about 25 mouse tissues in different developmental stages. Now this is a very different kind of comparison because you have the problem, okay, the challenge that you have different species, you have tissues. Remember yesterday we talked about the heterogeneity of tissues, and compared to humans' cell lines, many of which are transformed. All right? So any difference you see between those two, all right, you don't really know what the origin of that difference is. So, what do you do with this kind of comparison? What we understood was, though, if there were similarities that were maintained between these very different sets of experimental subjects, then that must be something that translates over evolution and must translate over the type of cell that's being examined. Right? So that initially gave us the idea that perhaps through this method we could come up with a very principled method of identifying genes that serve the function of maintenance and survival for the cell, right? And do that in a species and cell type independent manner. So these 18 cell lines in the human and 25 cell lines, and these 25 cell lines in mouse serve the function of giving the emphasis on looking at the similarities of these samples, and also gave us the opportunity to understand how variable were these genes that are similar in their expression level between mouse and human and different cell types. So not only could we identify the genes, we could also identify the range of variation that actually is occurring there. And that immediately told us something interesting, because almost all evaluations of this type serve to be based on the absolute level of expression. So does this gene express at 20 copies per cell? This gene is expressed at 10 copies per cell, or genes that are expressed at 100 versus 10. Those levels of expression have typically been used to denote a significant change or alteration in levels of expression. But this approach basically suggests that what you can do is not worry about the absolute levels of expression. They could be very different. But during the course of the two conditions or three conditions you're looking at, how much variation did that consistent set of genes have? So is there a constraint on the level of variation so that it is not the absolute level? Two genes can be housekeeping and have very different levels of expression, but they will not change their levels of expression over any condition beyond a certain range. So they're constrained in their variation. And that is sort of a new way in which to make this kind of expression monitoring comparison, in not worrying about the levels of expression, but how wide the range can they vary. So in these experiments, we had two biological replicates. And we took poly plus RNA extracted from each sample to make Illumina libraries using PAIR-100 at about 400 million reads per replicate. That's a very, very deep coverage within each cell line. Each replicate has almost 1 billion reads in each experiment. So that gives us the opportunity to look very deeply, all right, and in a very consistent fashion, because now you're not looking at 25 reads, or 30 reads for a particular gene, all right, or 100 reads per particular gene. With this depth, you can have very large numbers and therefore be more statistically confident about the differences you see. So when we made this first set of comparisons, we compared the dynamic range of expression, this variation range, in levels of mouse and human ortholog genes. So what we wanted to do is basically identify what genes could be comparable, right, in kind of an evolutionary fashion. So we looked at the orthologs in each species. So the dynamic range of each gene expression in a cell line or tissue sample can be up to about 6 orders of magnitude. So as we talked about yesterday, if you remember, we looked at that scatter plot separated between nucleus and cytosol, and we saw that protein coding genes had a centroid in the cytoplasm, but its distribution of range for protein coding was about 4 orders of magnitude. If you add to that the annotated non-coding transcripts and the new predicted non-coding transcripts, that was 6 orders of magnitude. And that's what we see here when we look at the differences between mouse and human, that in fact human and mouse have a dynamic range of about 6 orders of magnitude in both species. Now, when we looked at the number of expressed orthologs and primarily protein coding, but there's also non-coding transcripts in here also, we see that the mouse protein coding genes numbered about 20,000 in the samples that we had, in the 18 different, or 30 some odd different tissue samples that we had. And the human in total had about 18,000 protein coding transcripts. If you look at the expressed ortholog genes in human and mouse that are expressed in both human and mouse, there are about 15,000 orthologs, right, protein coding orthologs that are expressed in these experiments, in these tissues, in these two different species. If you add orthologs from six other species, mouse, macaque, rat, chicken, cow, there are about 6,000 of orthologs that are expressed in both mouse and in human. So there is about 6,000 genes that cross very large evolutionary space that are also seen in the mouse and human being expressed. All right? So that's the sort of characterization of what we're seeing in our general expression patterns. Now, one of the things we wanted to look at was the two-dimensional plots of the mean of expression versus the dynamic range of mouse versus human. So in this case, we took the expression mean in all tissues or all cell lines. We took that mean, right? And then we looked at and we plotted that against the variation, all right, in mouse and human for that particular gene, all right? And so what you have is this two-dimensional plot, and you have two forms of it here shown. One is that you have the variation that you see, and you have the mean expression level in log base 10. And so what you see here is that there are sort of two different centroids in this distribution, the seeds. One that has lower variation and one that has higher variation. And you see it here in this plot when you look at the density versus the range of expression. And what you see is, again, sort of two plots, two different kinds of populations showing up when you compare the two. One that has much lower variation than another group that has higher variation. And this is a more simplified view, that the dynamic range of gene expression across multiple cell types and two very different species has a bimodal distribution identifying two populations of genes with constrained and unconstrained levels of expression. And you can see that what is happening here is that the separation point of these two populations is about 2 logs of variation between the two populations, which means that there is a population that will not vary beyond two orders of magnitude, all right? Whereas the entire population, all right, and including the protein coding genes, will have at least a 6 log difference in their variation. And so that gives us a sense that there is a population whose levels of variation are constrained. That means they're regulated in a fashion that does not allow them, all right, to go and have extreme differences in the cell, irrespective of the species or cell type. Now, the number of constrained orthologous protein coding genes in the 6 different species looks like this. There is the total number of mouse expressed genes, as we indicated before, was about 20,000, 18,000 human expressed genes. And remember, we had about 5,000 orthologous genes, about 6,000 orthologous genes across 6 different species. And we can see, remember, that the genes that overlap between just mouse and human that were orthologous were about 15,000. Now, if you look and superimpose how much of these are constrained between mouse and human and between the 6 species, you can see that, in fact, there are about almost 2,500 genes across a very wide evolutionary level whose expression will not vary right, across across many different tissue types and many different species. This is important because this is about it's about 1/6 or a little less than 1/6 of the population of the total human gene expression, and about 10% of the total mouse expression. So that there's actually a fairly large number and consistent number of genes, depending on the species, the number of species you're comparing, that has this constraint on their variation. And they represent, although not a majority of the genes being seen as expressed, they represent a minority, but consistent minority of constrained expressed genes. One of the striking things that we noted is that we looked at the amount of RNA that was being expressed in mouse in the constrained versus unconstrained class. And what we see is that approximately 40% of the mass of RNA, that is to say the number of RNA molecules, 40% of that molecule is attributed to 17% of all of the annotated genes. And the RNA output is smaller for less differentiated cells. So that what we see is that we have a population of the proportion of cells that are expressed in mouse and in human, right? And the critical mass of genes that are expressed are in this constrained class, so that most of the RNA that's in this 25% or 10% is actually being made for the cell from that class. So although they're constrained, they're making most of the RNA that's the cell is populating the cell. And this explains this plot, which is that if you look at a correlation of expression in mouse and human across their entire genome, right, and just look at the gross pattern of expression in bins of about 1 million bases. So if you take 1 million bases, all right, and ask what's the level of expression in that 1 million, that window, right, and you do that across the 2 genomes, what you see is, and importantly, the genomes are aligned in orthologous fashion so that we have the same genes, even though they're on different chromosomes. That you see this pattern that is reminiscent and fairly correlated. If you make this correlation between the average expression levels in mouse and human across the genome, you have a correlation coefficient of almost 7.7. And if you align the alignable intergenic regions, again, this region's where most of the non-coding RNAs are coming from, you have a correlation of about 0.4. All right? So that basically says that this variation that we're seeing, and what's driving the comparison between mouse and human in this thing that's being quite similar, is coming from these genes that are being constrained. And because the mass of RNA is the most of the RNA that you're measuring in terms of their levels of expression. So, and this is basically a plot of of what we just saw, of the variation of mouse and human and the RPKM values in the human versus mouse. And what's in red are the constrained class. And you can see they are the most reproducible and correlative between the two different species. This is quite striking because in the end, what it means is that you now have a method whereby you can go into an experiment and instead of guessing based on their levels of expression, what are the housekeeping genes, identify those genes, set them aside, and then focus on the other genes as being the ones that are capable of being most responsive to the conditions that are changing for the cell that's being analyzed. So it's an interesting story because our intention was not to do this originally. Our intention was to understand the similarities between mouse and human and what regulatory elements were governing that. And instead, what we saw was to concentrate on the things that were consistently being expressed, as defined by their variation being constrained. And I think this method allows people to really focus on the genes that are truly responding to the changes in development or the environment that the cells are in. So I'm going to thank the people who did a lot of this work, from Roger Guido's lab in Barcelona, in the CRG in Barcelona, my laboratory, primarily Sarah DeVoldi and Julian Lagarde and Carrie Davis and Alex Logan in my group. They were the main drivers in this project, so I want to thank them. Okay, so does anybody have any questions on this topic? Yes.
I think that's an interesting topic. So for differential housekeeping gene and also for gene expression, usually we should think several levels things. For example, for one cell, for one type of cell, we think about different gene, different gene expression. From the cell type, one cell to another cell, one cell to another cell, we want to know the same genes, the expression level is changing or not. And the third, we should think that the overall expression of one cell type and another cell type. I think that I have remembered one paper published on cell. They also talk about the housekeeping gene. They think that for the tumor cell, the overall gene expression is much higher, and the normal cell is lower. So if we compare this gene expression, because tumor cell, even the housekeeping gene is also highly expressed, and the normal cell is not highly expressed. So if you use this to compare that some gene in tumor is highly expressed and the normal cell is lower, because the overall expression is higher, so this may be not true. So they use some single molecule to check that to definition. So for your result, it seems that you try to find just as a real housekeeping gene. This is not just between the cell type, also between the species.
Right.
The thing that if this gene is always a similar expression, but for the tumor cell, maybe the overall expression it is higher. So in this case, that gene even the same, but in that cell, maybe it means that it is downregulated.
Yeah.
So how do you?
So let me just comment on that, because this is, what we just said here is not usually the way people think about this. It is not the absolute level. It's not the amount of RNA, that's in one cell versus another. It's the change. We're looking at the values of the change of expression in the two conditions, right? That value, that variance, right, is really determines whether you can say this gene belongs to the class that is constrained and is maintaining some function within a range. Whereas these other genes that look like they have quite considerable variation across the two conditions, those are genes that are the most likely to be affected by whatever is the causative agent that you're trying to study, the transformation or development or something, or the environment. So it is this variation that we should look at, not necessarily the levels of expression, because the levels of expression can hide a lot of information. You can see a very large change in levels of expression in terms of the proportions, all right, or in their absolute mass amounts. But in fact, that variation, all right, could be quite small for the two genes. So you may have 1,000 copies, and it goes to triples that to 3,000 copies, all right. But the fold change, all right, is what's the difference, because that difference in the fold change means that it's not only changing the total number of copies of RNA, it's changing in the regulation that gives rise to that. Because if you allow the variation to be constrained, then the variation has to be tighter and more tightly controlled, and therefore less subject to the state that you're in. Right? So I think that's usually what we're trying to understand is how was the regulation of that gene, of those genes affected by this condition? Was the control lost? And what I'm saying is that definition of whether it was lost or not doesn't come from the absolute number. It comes from the variation that's there. Okay. It's a subtle, but I think important thing to keep in mind. It's not the absolute number of molecules, right, that's changing. It's the variation between the two states that you're looking at. Yes?
So when we study the transcription regulator, such as the epigenetic modifier, we always like knock out or knock down this transcription regulator and assuming that this housekeeping gene is not changing, like GAPDH to late actin or histone. But we're still wondering whether it's accurate when we study the transcription factor and change that factor level, and does this housekeeping genes never change or?
Yeah. So in the case where you have directed variability introduced to the cell through exogenous means, all right, or even in the case where you have natural mutations, affecting a regulatory element, that one gene may in fact change not only its absolute number, but its variance also. But what I'm trying to point to here is that there are almost depending on the number of species that you're looking at, there's almost 2,500 genes whose variation should not be much greater than roughly 2 logs, right, in the 2 states. That's a list. That's a real list of genes, right? That's not something you have to figure out in the experiment. And if you see an effect that brings that variation outside that range, right, then you know that this is a very serious mutation. It gives a sense that a gene that's much more tightly controlled than two-thirds of all the other genes has now lost its control of that. And that has a very definitive start. One of the things that I didn't mention, thank you for reminding me, one of the things I didn't mention is that if you look at genetic diseases, human genetic diseases, the ones that you see, the genes that you see affected from the germline that are passed along from generation to generation, those germline mutations tend to be in these constrained genes, right? And the ones that are somatic, that occur spontaneously as a cause of the disease, those tend to be in the ones that are in unconstrained class. So the most serious ones, it's a comparison that you don't really can't make black and white of, but the fact that you lose control of these housekeeping genes means that there is something that should not be varying quite in a very large fashion is now actually lost and therefore has consequences over the life cycle of that cell. So I think there is an important also caveat in this, that these kinds of genes also tell you the importance of whether a mutation is happening there or not.
So where can I get this range of this constantly expressed?
They're in this paper that we published in Nature Communications last year.
Oh, that's great. And so how do we calculate whether the housekeeping genes in our condition is within the range? I mean, so we have to do the RNA-seq to... Yeah,
so it is basically, you need at least 2 conditions to compare, right? And you need to have reasonably statistically sound numbers to see whether the variation you're seeing is statistically significant or not. And that's sort of the minimum required. This is one of the values of having done such a deep sequence for each sample, almost 1 billion sequences for the 2 replicates. That gives us some very sound numbers to see what the real variance is in these 2 genes.
Thank you.
Okay. Yes.
Why did you rule out the non-coding genes?
They weren't ruled out. It's just that if we go back to this, let's see. If you go back to this slide, what you see here is that if you look at the intergenic regions that are alignable between the 2 species, you still get this very strong correlation in the levels of expression and the variance that you see in those regions. Almost all of the RNAs that are being expressed in those regions are the non-coding transcripts, the long intergenic non-coding transcripts. Right? And so the reason we wanted to do that was we could align those right? Because there are very few isoforms for those in those regions. So we know exactly which RNAs are being expressed. Whereas if you look at long non-coding RNAs that overlap annotated protein coding genes, right, then it would be very difficult to know whether you're actually seeing evidence of the long non-coding transcript or the protein coding transcript that's influencing this. So in these numbers, we focused on the protein coding genes, but if we did the same analysis for the non-coding genes, we would see the same kind of conditions, this kind of constraints it. So your point is well taken. There is a collection of non-coding transcripts that act as if it's a housekeeping collection.
In this case, that housekeeping gene is a complete gene or housekeeping gene, not
protein coding genes, but some of no coding gene also can be
Yes.
housekeeping complete genes.
Yes.
What's the, for example, what's the percentage between the protein coding gene and the no coding gene in that totally constrained that gene?
Oh, it's vastly biased towards the coding transcripts.
Most of it should be
Most of the non-coding are in this more variable class.
Okay.
By a very large factor. Most tend to know. Okay, any other questions? Any other comments? All right, let's move on. Okay, so now I'm going to ask you to think about expanding the horizons of what we traditionally think about when we think about RNA expression, right, and the roles for RNA. Almost all of the literature that you're familiar with, all right, until very recently, concerned itself with the operation of protein coding and non-coding transcripts, right, within the boundaries of the cell. There are very few topics that covered anything that was being released outside the cell, and certainly RNA was not one of them. Right? And more traditionally, that was a domain left to protein coding elements. More recently, we've become more appreciative of the fact that there are elements that are being released to the cell in a packaged form, that is to say, in a protected form, right? Either in a vesicle or bound with protein sufficiently to protect the RNA or other element that's being bound from being degraded by bound elements outside the cell. Now, many of you are probably familiar with the concept that most of this material that was thought to be jettisoned from cells was thought to be basically a garbage disposal unit for cells. Things that had were not being needed by the cell, things that needed to be recycled and degraded into more elemental forms, were thought to be released from the cell for the purpose of getting, not clogging up the cell with these elements that were thought to be not as functional. But more recently, this concept has been re-examined and redefined. So we, as scientists, basically, many of us do this very simple kind of experiment. This is an example of an experiment, not really what we are talking about. What we want to know is that if we take two conditions, in this case here we have a collection of condition 1 versus condition 2. We're measuring the levels of expression in condition 1 versus condition 2, and each dot is a expression level for a given gene in condition 1 versus condition 2 for a given gene. And then we run these in sort of replica fashion. Here we have 3 replicates over 3 time points. So when we do that, we want to see whether there's any comparative differences, for example, between one type of cell and another type of cell over the same 3 conditions. And this, for example, is a time course from time 0 to time 24, and we have 2 different cell types. And we want to know, are there patterns of expression that are similar between these 2 different cell types? And in this kind of study, we can see that, in fact, there's almost completely the opposite. We start with a relatively similar kind of pattern, and then we move to something that is relatively highly expressed versus something that's relatively lowly expressed after 2 hours. And then you have even a greater difference here after 24 hours. And this is the kind of general experiment over this time course that we do. It's just a general way we've been looking at differential expression for a long time. The question then becomes that can we see this effect, between condition 1 and condition 2, when we expose different cell types over time or over different environmental conditions, if the stimulating environmental condition is elements that are being released from a cell, right? And those elements that we can focus on very specifically are those elements that are put into small vesicles that we can find outside within the cell media or in the blood or in some bodily fluid. And so do these vesicles contribute to that pattern that we see? In other words, what we see the response within the cell, is some of that caused by the fact that cells are releasing material around them and helping to change the environment. So it's not just two cell types we're looking at. We're looking at the differences in cells talking to each other via these vesicles. If they are, what are the causative agents within the vesicles? Do they include RNA? Are they active agents found in the vesicles the same structure as in the cell of origin? Is the element that the cell has packaged into the vesicle exactly the same as you would have had in the cell it came from? Or has it been changed explicitly for the purpose of being put into a package and sent out of the cell? And finally, what do the active agents do in the recipient cell? What is the phenotype of these active agents? So when we look at this kind of experiment here, what we're looking at is the response of a cell. And we call it the difference between cell type 1 and cell type 2. And we think of it only in terms of what's going on inside the cell. What is the mechanisms inside the cell doing? Because one is a cell type 1 and one is cell type 2 over a time course. But the other factor, which we don't actually consider, is that cell sitting in, that cell type 1 sitting in a culture vesicle or in a human being, right, and being monitored over time, is also being influenced by elements outside of the cell. And those elements outside the cell could actually be contributing to some of these changes in level of expression. And what are they? Are there any at all? And what do they do? And what are they? Right? So the first task is to find an example where we can definitively show that we have a cell type, right? We gather this externally released material from somewhere else and expose it to that cell and ask, do we see something different about that cell? Can we see not only a change in expression, but can we see a different phenotype? And then we can ask, what's in that vesicle that has caused this? Is it a protein? Is it a lipid? Is it a carbohydrate? Is it RNA? Is it DNA? What is in there that has caused this change? Right? And that will tell us whether this experiment has elements in it that in fact have been influenced by the external environment in which it's been placed, not only over time. Okay, so these vesicles, I'm wondering, before I launch into this, do you guys want to take a 10-minute break? Because it's almost an hour. I can keep going, but it's up to you. Okay, 10 minutes. All right. So these vesicles have actually a relatively short history. Right? They were discovered, materials have always been found in culture media outside of the cells. And that's very easy to prove because all people had to do was take the cells, spin the cells out at a very low level of centrifugal force, all right, and then take the remaining things that are cell-free, right, and spin that very hard, right? And what you see, very simply, is a pellet at the bottom. It's not a very big pellet, but there's material in there, right? And that was just thought to be degraded cells, lyse cells, and the material that comes that are no longer part of a whole cell. Right? And that was sort of underlying the presumption that this is a garbage can area, that garbage was just dumped out there. But more recently, in the 80s, what was observed was that reticulocytes, right, had these large vesicles within them. Right? And this is a paper from Harding's group in 1983, these EM micrographs. These had large vesicles within the given cell type. These are blood cells. And these large multivesicular elements had what looked to be smaller vesicles lodged within them. And if these vesicles were in a condition where you feed ferritin, an iron-containing protein, a very dense containing protein, and you do an EM micrograph, you see that this ferritin gets brought into these large vesicles and are associated with these little vesicles. Under scanning EM, you can see the vesicles actually in these membrane pits that are present. And eventually, these vesicles are brought to the surface of the cell, and with these attached ferritin particles, and they're released outside the cell. And these small little vesicles are now free to be detached outside. You can see it here in Johnstone's paper in 1985, the exact same thing in a more dramatic fashion. And so what was thought, in summary, was that ferritin was brought in, it was attached to the elements inside these bodies, or it was brought in and attached to smaller elements inside these large vesicular bodies and then released. So there were two possible pathways that could go. One where it goes in and is specifically sent out, and one was associated with small vesicle bodies within it. And that was the original observation that gave rise to the concept that these bodies may be very specifically made and have a pathway of their synthesis. So what we know about this pathway as of today is that you have a secreting cell, and it has two pathways in which it releases bodies. One is that the outside membrane basically blebs out, all right, and then elements inside of the cell get trapped in these blebs and are released from into the environment. And the second is that there is a reticular body here that basically has an internalization of that membrane into the cell. So the outer wall of the cell, this outer wall here, is internalized inside of the cell, and a new membrane is formed, an inside-out membrane from what it was before. This is an important concept, because why that's important is because the outer surface is what's exposed to what's in the cell, right? And now it becomes the inner contents of that vesicle, right? So there's a means in which to put elements from within the cytoplasm into a vesicle and then eventually released outside into the environment. This kind of pathway of vesicle biogenesis and release led us to the concept, or led early researchers to the concept that this might be a way in which to get different kinds of molecules into this. If you could get proteins, you could clearly get RNA, you could clearly get carbohydrates and other modified elements into these vesicles. And what has sort of been, as we discussed yesterday, this sort of need for us humans to catalog things, to organize them into lists, to put them into different categories, took over, right? And what happened was that although Harding was identified these in 1983, historically, these vesicles were cataloged and organized according to their size, because it was the easiest thing to do, right? And their size was seen as something that was quite small, between 30 nanometers and 100 nanometers, right? Things that are sort of intermediate, and things which are quite large, a micron or more. Now, this is in some ways kind of silly, but nevertheless, it gave us a way to begin to segregating the populations. And that these small vesicles were called exosomes, the mid-sized vesicles were called microvesicles, and the large vesicles, which later became very clear, it was almost the size of a cell, whereas the residue of lyse cells, otherwise known as apoptotic bodies. You can see at this level, you're almost at the diameter of whole cells in places. But the reality is that these extracellular vesicles are composed of a spectrum of sizes. Right? There's a continuum, right, from about 20 nanometers all the way up to about a micron. Right? And that continuum, right, has no real boundary. We don't understand how to make a boundary between one type of vesicle and another. In fact, we don't even know whether there's one type of vesicle, as we'll see in a second. Is there something different about things that are as cargo elements in this versus things that are in cargo element in that? Are there different kinds of molecules that are different? More importantly, and clearly not understood, is that if you have two of these, do they have the same contents? Right? If you have two small vesicles coming from the same cell, do they have the same contents? Right? And finally, we know that these vesicles are a difference in size. Are there more of the smaller ones than the bigger ones? Right? No one really knows. Right? But you'll see the partial answer to this in a second. Now, we became interested in this because I had a very, I would say, brave, that's the right word, brave graduate student. Right? He came to my lab 4 or 5 years ago, and he said, I want to look to see if an element in these vesicles, right, which at the time we knew there was RNA in these vesicles, whether these RNAs are capable of actually causing a phenotype. Do they have functional properties, right, that are associated with these vesicles? His name was Sudhito Chakravarti, and he went to work. He wanted to work on this, and I said to him, "This is crazy. I mean, we don't have any experience in isolating these vesicles. You're going to have to learn how to do micrographs, which we didn't do. And there's a whole list of reasons." And he very quietly sat there and looked at me, and he said, "Yes, but I want to do this." And so he was very brave and very stubborn, and he took on this project. One of the first things he had to do was to find a method where we could isolate and highly enrich these vesicles, right? And separate the mid-sized ones, right, the things that are called microvesicles, right, from the exosomes, right? And we had to do it in a way in which the yields, that is to say, the amount of material isolated from 1 million cells, for example, right, was enough to actually do RNA-seq experiments and to do other kinds of experiments in that material, which is not a trivial matter. And finally, he had to find a method which was relatively non-laborious, right? And I'll explain what that means in a minute, and also cost-effective, not really effective. And I say that because at the same time, there were many companies jumping into this field, all right, because of the literature that was suggesting they would be good biomarkers, all right, for different kinds of diseases. So many companies jumped into this field, and the first thing they did is build kits, all right, to basically isolate these vesicles. And there were very different kinds of kits. This plot here is basically a demonstration of a precipitation method and with a kit, and then basically your pellet is enriched in these small exosomes. There are methods which have been in the literature for a very long time involving multiple ultracentrifugations to separate the large vesicles from small vesicles, right? But this this method of precipitation is very expensive, and this method here takes a few days to do and a lot of labor, a lot of manual intervention. And then there's another method, another commercial method made up for filtration, and where you basically pass the supernatant from the cell through a nano-sized pores and isolate the vesicles in that fashion. But this is also expensive, but more importantly, this is fraught with the problem that if the pores get clogged, all right, then your yields drop tremendously, and so it's not very efficient in gathering these vesicles. So instead, he looked at all of these different methods and basically came up with a sort of mixed method in which he combined the filtration method and the ultracentrifugation method, and then colored it with the small volumes here. The problem with the cost here is that you have very large volumes of the culture medium, in which you have to add a lot of these reagents, right, to in order to get a pellet from. Here, if you have, after you filter, and you have small volumes, and that doesn't require much of the material, and is much less expensive. Anyway, he used this mixed method to isolate what we think is something in the order of almost a million-fold enrichment of these vesicles from the supernatant culture medium. The next step was to basically determine whether the isolated vesicles here were truly belong to the vesicles and not to the cells themselves that are there. And so what we did was do Western blot. He did Western blot analysis of using Alexin CD71 and TSG 101, which were known membrane surface markers from these vesicles, and compare them to the whole cell extract, or Western extract. And you can see that these have highly enriched contents of these markers. And then all the internal cell markers that should be excluded from here clearly show that there's a clear separation of the membranes that we see in the vesicles versus those that are in the cell. The next thing that we did was to actually take electron micrographs of the vesicles, of the purified vesicles, and also stain them with antibodies containing gold particles, right, and show that the targets for the antibodies were on the external portion of these membranes that we were isolated. And that indicated to us that we didn't see other kinds of structures in this preparation that could be confounding in the analysis that we were going to do. And we looked at the concentration of the EVs and the size versus the size of the EVs, the number versus the size of the EVs. And we saw a sort of median size of about 200 nanometers as being the median size of the vesicles. And finally, we looked at the contents of the RNAs, the signal intensity, and the size of the RNA that we could extract from there. And what we noticed was that clearly the majority of the RNAs that we see are smaller than 200 nucleotides, and that there were virtually, these are markers that in the preparation, there are virtually no long RNAs that are in the vesicles that we had seen. And so you can think of it this way, that these are reservoirs of material, and these reservoirs, the question was whether these reservoirs were containing functional RNAs. Okay? And so now I'm going to tell you about the story about how we set about testing whether they're functional RNAs or not. So why don't we take a break here, and then I'll get into the biology that we have. Okay? Back. Okay, let's get started, okay? All right, so we discussed the fact that we had a method to isolate exosomes, and we also had a method by which we could measure the enrichment factor of the exosomes that in the vesicles that we have. I keep using this term. The field has moved has been trying to move away from this idea of calling things microvesicles or exosomes, depending on the size, and sort of accept the fact that there's a continuum there, right? And so all of these elements are now being called just extracellular vesicles, EVs, right, as a general principle, as a general name, right, without trying to make a distinction based on size. Anyway, so we have these characteristics done, and we had to then figure out ways in which to evaluate whether the vesicles had a biological influence on cells around them, either as isolated vesicles being used to treat cells or from one cell to another. And the way we did this was to utilize a well-established experimental condition called a trans-well assay, in which an upper compartment is placed into the culture media, and that upper compartment is separated from the lower one by these micropores, which have about a 200 nanometer diameter aperture. And then either you can take cells of one type and expose it to a second type, or take cells, take these cells, put them on the bottom compartment, and put the exosomes up here, isolated exosomes, and look for change effects in the lower compartment. In either way, this is the standard way we did these kinds of experiments. So, when we isolated these different exosomes, and the fact that we could see, as the literature had taught us, RNA present in them, and that the RNA that we had seen in them was actually not very large, not very long, up to about 200 to 300 nucleotides in length. We wanted to know what kinds of RNAs were there. Now, many experiments have been done like this. There must be at least 100 papers in the literature where vesicles have been isolated and or claimed to be isolated, and then identified as having these kinds of RNAs in them or not, and then compare it to the cells that they were derived from. The difference in this experiment was that we saw no long RNA. There are claims in the literature that full-length messenger RNA molecules were present in these things. We have never seen any evidence of that. What we see is fragments of long RNAs, all right? And often that is enough for people to say that the long RNA is present. But we have never seen this these kinds of long RNAs. Now, I have to be very specific about this. We isolate these vesicles in a very specific fashion, and clearly we bias towards the smaller size vesicles. It may well be that in a larger vesicle you see these elements, but these long non-coding RNAs, but we have never seen them as yet. So what we did was to take a look at two cell types, a primary cell type, BJ, and analyze the RNA that's present, the small RNAs, the short RNAs that are present in the BJ host cell primary cell line, and the EVs, the vesicles that come from these BJ host cells, right? And then compare them to the whole cell short RNA content in K562, a lymphoid cell line, and it's a transformed cell line, and compare it to the vesicles that are coming out from there. Now, it is quite striking that the different kinds of short RNAs that we observe mainly come from elements that look like they are microRNAs, tRNAs, and fragments of protein coding elements that are there. So that the comparison between the whole cell of the primary cell line versus the EV, the most dramatic difference was in this area of RNAs called the miscellaneous class of RNAs, which were less than 1% of the total number of reads observed in the whole cell. But in the EVs, were almost 40% of the total number of reads. So you have this almost well over 100-fold increase, all right, in the content of this particular RNA in EVs compared to the donor cell. Similarly, we see this same enrichment in the cancer cell, where the enrichment is even greater. It's almost twice as much and again, focused on this class of miscellaneous RNAs. This miscellaneous class of RNAs is the class of RNAs that is sort of a catch-all class, as we discussed yesterday, containing Y-RNAs, SCA RNAs, and Volt RNAs. This enrichment that we see here is notable because of the Y-RNAs that you see, most of the miscellaneous RNAs are Y5. So most of that enrichment comes from the selection of one kind of RNA from the cell, right? And that RNA is one member of a family of RNAs which belong to this miscellaneous class. These Y-RNAs have been characterized for many years. They are Pol3 transcripts, right? Which means they are not polyadenylated. Y3 is the most conserved family member, and Y5 is the least conserved. And you can see the very tight hairpin structures that can be drawn, modeled for each of these, for 1, 3, 4, and 5. The contents of these RNAs are seen to interact with some very specific proteins, namely Rho60 and LOC. And the binding sites for those proteins are seen at the 3' ends and in the hairpin of the RNA. We have here Y1 RNA as an example. They see these RNAs in 2008 as required for increased proliferation of cancer cells. That's been a published set of results. And necessary but not sufficient for DNA replication. And that is perhaps the most controversial and yet probably the most consistent with the story I'm about to tell you, that somehow it's involved in replication of cells. And then it's also been stated as it's part of the RNA processing QC of RNAs that have not defolded correctly and have been shepherded out the cell. So again, human have 4 members of the family. Mouse has only 2, and C. elegans has 1 member of the family. But so evolutionarily, this class of RNAs go very deeply into evolutionary history. Right, so they've been conserved for a very long period of time. When we looked at these RNAs, so we saw this increase in high enrichment of RNA Y5. We asked the question, how much RNA Y5 was there, and what gave rise to the RNA. When we looked at the sequences that we obtained from the EVs, we noticed a very striking phenomenon. What we noticed was that if you looked at the 5' ends of the Y5 molecule, you see 2 prominent 5' ends, one at nucleotide 1 and one at nucleotide 52. We saw 3 generally prominent regions of 3' ends, one at nucleotide 23, paired ones at 29 and 31, and the terminal one at 83. I neglected to mention that the RNY5 is 83 nucleotides long, so the parent Pol3 transcript is 83 nucleotides long. So we looked at these sequences and saw that what we find inside of the EVs, all right, was a full-length version, a version that started at 1 and ended at 23, a version that started at 1 and ended at 29 or 31, and another version that started at 52 and ended at 83. Right? And if you look at the model of RNY5, what you see here is this molecule here, this 31 nucleotide molecule, is the dominant form that you see inside of the EVs. And is the most prominent part of the molecule that you see. You see the 52 to 53, and that basically starts on the other side of this loop out and comes around to the terminus that we see here. So this are 2 halves of this molecule, suggesting that this open single-stranded region is a site for cleavage of this molecule. We then went about and asked the question of how much RNA was in the cell and versus in both the whole cell and the EVs in the primary cell versus the cancer cell. So when these are markers at 83, 31, and 23 nucleotides. And what we saw was that in the primary cell, the BJ cell, you see only basically 83 mer, and not at very high levels. There's very little enrichment of the 83 mer in the northern from a whole cell. In the exosome, though, we see this prominent product being made, and this prominent product was not 31 nucleotides, which is this one here, this marker here, but 23. So in vitro, unlike in vivo, where the prominent product is 23 nucleotides. We see the same thing in the transformed cell, where we now see a relatively large amount of RNY5 as a whole 83 nucleotide product, a much more reduced version of it in the exosome, the whole transcript model, but a much larger amounts of the 23 and 24 nucleotide product. This indicated to us that this cleavage, although 23 is present in the whole cell and the EVs, this RNA product is preferred when you're outside of the cell than inside of the cell, inside the EVs. So indicating that there's something protecting the 31, this site here, as opposed to the 23 nucleotide site in this single-stranded region, which is a more prominent site for cleavage. It's important to note that the literature had noted that these 8 nucleotides in both the 23 and the 31 nucleotide fragments was implicated in the 83 nucleotide as being essential, required, but not sufficient for DNA replication. So the function of the 83 nucleotide to help set up replication initiation complexes based on the 83 nucleotide required these 8 nucleotides to be present. All right? So this will become important in a second when we talk about the processed versions rather than the whole 83 nucleotide molecule. The next question that Sivito was interested in was the question of where did the activity to give rise to these fragments come from? Were they actually in the whole cell, all right, and then packaged with the RNA and then sent out there? Or were they actually only in the EVs, all right? And all that processing happened within the EV specifically. And so you can see the results here. This is the 83 nucleotide parent molecule. These are the size markers, 23 and 31. And if we take the activity from the whole cell, so we make a protein extract from the whole cell, right, and we treat the 83 nucleotides, we basically see no processing of that 83 nucleotide whatsoever. If we take extracts from the EVs, right, from the EVs, and treat the whole cell 83 nucleotide marker, we see this dominant 23 nucleotide and a pre-step getting to 23 at about 45 or 46 nucleotides. So this processing happens explicitly in the EV compartment and not in the whole cells. The activity of this processing is actually dependent on these 8 nucleotides. If you scramble these 8 nucleotides, right, and then ask for the processing to happen, what you see is that when you have when you have a scrambled version, all right, you basically have no. When you have no you have a scrambled version with no extract, right? You maintain the 31 nucleotide precursor, and then if you scramble the version and then extract, you degrade that RNA down to something less than 5 or 10 nucleotides. If we scramble the whole molecule, then we basically see nothing more than degradation products from this material. So that we see that this recognition for this cleavage is resident in the EV and requires the 8 nucleotides that are present. If we monitor you monitor the speed at which this cleavage occurs at, what you notice is that at time 0, at 83 nucleotides, you begin to build up this 23 nucleotide extract, and this extract has very little activity in terms of the whole cell. In the exosome, you see this extract degrade 83 nucleotides specifically for the 23 nucleotides present. So this indicates that very early on and very rapidly you have this cleavage event happening. The RNA that we see in the cells can be visualized, can be sort of shown to be transferred from one cell to another by basically labeling the lipid membranes of the vesicles that are being transferred. So in this experiment, we take a transwell assay and have the K562 cells above and the BJ cells below, and we allow for gravity to bring the vesicles down to the recipient cells here. And what you see in a sort of a static assay is the presence of these vesicles beginning to form a perinuclear ring around the nucleus in the recipient cells. Remember, these dots are membranes of the vesicles. These vesicles have been labeled in the donor, from the donor cell and brought into the recipient cell. Now, in addition, you can see that cells release these vesicles in this fashion. That's a little dark, I guess. Trying to. It's pretty hard to see this. There is a vesicle right here, which I can see in my screen here, but not so well here. There's a vesicle at this point, which has been released from this whole cell. And when it goes and enters the recipient cell, you see these vesicles within the membrane. This is the nucleus of the cell, containing these vesicles inside of it. But you can see it enter the cell and indicate that the transfer from one cell type into the other is clearly possible. Now, if you do the same experiment and now take the cancer cells in the upper domain and the primary cells in the lower domain, and you ask, do you see a phenotype happen? This is what you get. If you take cancer cells and treat the cancer cells on the top and cancer cells on the bottom, and let the EVs transfer from one cancer cell type to another, you see that there's virtually no difference in terms of the viability of the cancer cells. None of them enter a phase of cell death for the cancer cells. However, if you take EVs or total RNA from EVs of cancer cells and you treat the primary cells, what happens is that you see this response in terms of overall cell increase in cell death with either the EV or the RNA from the EV. And so this experiment basically says that if you take the EV from the cancer cell, you treat a primary cell, the cell begins to die at a fairly rapid pace. If you take that EV from the cancer cell and extract total RNA from it, all right, and then treat the primary cells, those cells also die, just like for the EVs, indicating that the RNA that's in these EVs, all right, is part of the causative agent to promote cell death, specifically of the primary cells. If you make a synthetic version, all right, of the 31 nucleotide or the 23 nucleotide, so now instead of taking it from the EVs, you just synthesize that molecule. Importantly, this molecule will be unprotected at the 5' end or the 3' end and unmodified, or anywhere along its length. If you take that 31 nucleotide or 23 nucleotide synthetic version, it causes an increase, statistically important increase of cell death in all of the primary cells that we've looked at so far. And now we've looked at about half a dozen of these primary cells that, of very different lineages. If you increase the amount of the synthetic R1 of 31 or 23, you get this dose response, clearly indicating that the accumulation of this small RNA, all right, has an increasing response on the part of the recipient cells in terms of the phenotype of triggering cell death. If you have an intact 31 and 21 that cause cell death, a shuffled version of either of these two, shuffled meaning either the entire 31 mer is shuffled or just the 8 nucleotides, all right, then you lose that cell death phenomenon. And finally, cancer cells are not killed by any form of the 31 mer. So we can either give the intact version of the 31 mer or a scrambled version of the 31 mer, and basically that cell death phenotype is abrogated also. So now from these experiments, what we can see is that the active compound in the EVs, right, is a 31 or 23 nucleotide product, and that product is the result of cleavage that goes on inside the EVs, and you get the same kind of phenotypic response whether you're using a synthetic version of the RNA molecule or the whole EV or the total RNA extracted from the EVs. We have very clear evidence that RNY5 is the active agent by another experiment. In mouse, there is no RNY5, so it's as if it were a genetic knockout of RNY5. So there is no RNY5 genetically in the mouse. But if you take the recipient cell as HB4 and the donor cell K562, which is going to donate the RNY5, and you put them into that transwell assay, allow them to incubate, and then pick the mouse cells up, all right, that as the recipients, what you see is this increase of RNY5 sequences specifically in the mouse, all right, in about 12 hours, unambiguously indicating that RNY5 is entering the mouse via by vesicles coming from the human cell. That has some very important implications. Those implications are that since this comes from an entirely different species, all right, via an EV, all right, that the recognition of that EV, all right, is in fact not dependent, all right, on a donor-like receptor complex that is species-specific, right, which is something that is a bit puzzling, since there have been many published literature that suggests that targeting of EVs is part of the specificity of the EV. So that EVs made from one type of cell in a human prefer to go to other cell types to be taken up. At least in culture, what we see is that this is not the case. That there seems to be a general availability of EVs to enter into other cells, even if they're from a different species. So we have to be careful that this experiment is done in culture and is not done as an in vivo situation. So in summary, what we see is that primary cells, if they're exposed to exosomes coming from cancer cells, and we've looked at about 5 different cancer cell lines to confirm this, result is that the EVs cause cell death of these primary cells. The important thing about the cell death phenomenon, which you can see from this, is that in this titration, even at fairly high levels of the synthetic RNY5 fragment, that 31 bar, at most you'll see is about 40 to 50% cell death. Not all cells die. All right? Now, the reason for this is one of two possibilities. One, that there are resistance sublines in that cell line of primary cells. Or two, that the phenotype is dependent upon the cell cycle stage of the cell. If the cells are not dividing, for example, or they're in some sort of senescent state, or have reached senescence, then they may not be responsible for this. And we're sort of working on this now. So, but it's important to know that if you take a culture, at least in vitro, and treat them in these fashions, either with EVs or with the RNA itself, the response will not be for 100% of the cells. And it will be intriguing to know whether there's a resistant cell line, resistant subcell line in those groups that differentiate them in terms of response or not. Now, why do I think that might be a possibility? There is an intriguing set of papers from one of my colleagues at Cold Spring Harbor that noted that in the case of xenografts of human breast cancers, right, in mouse, right, if you put xenografts into a mouse, the fibroblasts of the breast cells begin to die as a result of this transplantation, much like we see in this culture. But they don't all die. There are some fibroblasts that seem to be providing supplement material for the xenograft tumors. That is to say, you can exogenously remove those fibroblasts, co-culture them with the breast tumor cells, and their rate of growth and their mobility status changes dramatically. If you take it from a mixed population of breast fibroblasts and put them into the same culture, you don't see as much dramatic, because many of them are dying as a result of the co-culture with the tumor cells. So the possibility, this gives rise to, again, speculation, very important, speculation on that there are two populations of primary cells in these co-culture experiments. Ones that are responsive to the cell death signal, and those which are not, because they are providing some kind of complement to the tumor cells themselves. And, but the other is a more trivial, namely it's dependent on the cell cycle that we're in. Okay, this, again, if you extract the total RNA from the exosomes, then you see the same kind of cell death. This is an important experiment also, because what you note in this is that the relative amount of RNY5 isolated in this, all right, is lower, all right, than if you took the purified 31 or 23. Remember, in that case, you have a homogeneous population where every molecule, all right, is potentially active. In this case, although RNY5, the fragment, is the dominant species of yRNA in there, it's not the dominant RNA, all right, in terms of mass. So you get the same level of response with the extracted RNA as you do with the 31 nucleotide purified ones, suggesting two things. One, that there's a plateau of response, even if you give it more active molecule, right? And number two is that the in vivo, right, it takes perhaps a very small amount of this molecule in the background of a large number of molecules to cause this phenotype. And that's actually could turn out to be very important. Okay, again, the synthetic version leads to cell death. Two things which I didn't tell you, all right? which we found out, is that if you synthesize the 31 nucleotide active fraction and you put it with its complement in the 83 mer, in other words, you make the second strand. And if we go back to the model, yeah. So if you make this 31 mer and you make this 53 mer, all right, and make it double-stranded, right, and you put it into the experiment, you aggregate the cell death phenotype. The cells don't respond. Even though the 31 nucleotide is present, right, its complement appears to aggregate the response. And as expected, if you put the full-length 83 mer and transfect it into primary cells, it has no phenotype. And we believe this is likely to due to the fact that this happens because it's, again, the 31 nucleotide containing the 8 nucleotide sequence that is important is now in a double-stranded form. And it's a single-stranded form that turns out to be an important aspect of this. Okay? Now, we're going to talk a little more about this phenomenon of the cell death and ask the question of what is it happens to the cell when it receives either the ES cell, the EVs, or the synthetic RNY5 31. Okay? And what I'm about to tell you is true for both. If you give the recipient cell the artificial 31 mer, or you give it the EVs from the cancer cells, all right, those cells respond in the same fashion by virtue of the fact that the TGF-beta pathway is immediately affected in these cells. These are all downregulated, and in addition, P300, c-MYC, and a variety of other RNAs are upregulated. What happens is that these eventually are used to control the cell cycle, that is to say, DNA replication in the host cells. And in doing so, the cell cycle is now unchecked, so cell cycle control is lost, and uncontrolled replication begins, triggering apoptosis in those recipient cells. So, consistent with the observation about the 83 mer being involved in establishment of DNA replication contents, the single-cell version, the 31 nucleotide, triggers uncontrolled replication here in a single-stranded form. And we think that this is a mechanism by which two proteins, some of the CDK proteins, are in fact the most affected by the RNY5. We don't know what the RNY5 is interacting with in this phenotype in order to cause this very pronounced and consistent repression of these genes in this pathway. So we don't know what the mediator is, but we now have a variety of mass spec candidates that we're looking at for this. And this is just a larger picture. And I bring it into here because there are also two microRNAs, miR17-92 and miR93, which are also highly upregulated and enriched in these vesicles. Right? And these turn out to be negative regulators of P10 and XX31. These microRNAs are massively upregulated to enhance the effect seen as the TGF-beta, TGF-beta pathway effect that we see here. All right, and I'm going to stop in a second or so, but I want to stop this section with the fact that in the exosomes, cancer exosomes that we've seen and analyzed, we have seen these microRNAs specifically as being upregulated by these folds, about 150-fold, 70-fold, 50-fold, and so forth. These have been significantly upregulated in the EVs that we've studied, and each one of them has had a very clear correlation with promoting cancer development in a variety of different cell types in cancer. So, well, the role of these microRNAs, I know, is a very prominent interest of many people, and even here. And it looks like that there's obviously more than one kind of functional molecule that may be operating in these EVs. Okay, so I think I'm going to stop here, and let's take another 5-minute break, and then I'll finish up with a discussion about another class of RNA molecules that are phenotypically important and come from EVs. Okay. Primarily at 3' ends in the whole cell. And then we have a large number of elements that are selecting against. There are very few 3' ends in the EVs, much more enriched in the whole cells. And then there's this population that have both the 3' ends of the gene in the whole cell and in the EVs. So it's very common to have 3' ends, right, present in the whole cell and in the EVs, all right, for these genes. Then there's a collection of genes which have 5' ends present, and in there we have this collection of genes that are present in the whole cell only, and then this collection, which is predominantly, all right, just the 5' end, with very little or selected against the amount of 5' ends in the EVs. So unlike this one, where when it's together, there are fairly similar ratios between both ends, the 5' ends look like they're being specifically enriched for a collection of genes, of tRNA genes. Now, what are those tRNA genes? If we look at histidine in the BJ cells, there are 440 tRNA genes out of 624 expressed. There are 3 histidine tRNA genes, all right? And if we look at this gene, which gives rise to a histidine transport vehicle, 78% is found in the whole cell. So of all the histidine genes that are being expressed, 78% come from this gene, and it's found in the whole cell. The other proportions in the whole cell are coming from this gene here, the roughly 30%. However, if you look at this gene, 96% of all of the histidine gene expression is coming from this one gene. It is going to the EVs. So, out of 3 genes that histidine uses as part of its translational function, it picks one, right, and almost the entirety to put into the EVs. And what it puts into the EVs is the 5' end. All right? Similarly for Lin, there are 7 genes that comprise genes that make tRNAs for Lin. This one here is the gene that's selected for specific export, its 5' export into EVs and out of the cell. The rest remain in the cell itself. This is a very complicated slide, and I'll walk you through it, but it's meant to tell you that this phenomenon I just showed you for 2 is happening globally in terms of the tRNA profiles that we see. So let's look at this. These are the tRNA gene families expressed in BJ cells. And this is the gene families. This column is the anticodons. All right, so obviously valine has 2 anticodons, serine has 3, and so forth. Okay? Now, so we're segregating the tRNAs based on whether they come from a specific gene family, or whether they are separated by their anticodons, okay? Now, if you look as we did before, for histidine, we can see that we saw that 98% of what goes into EVs came from one gene, and here it is, right? It's basically here it is, the blue. It's almost entirely designated for the EVs, and it's making up most of the RNA that's made in the cell, but it's being exported out. This is the gene that's made explicitly for cell consumption. And then there are, there's the glin, all right? And it is, most of it is coming from this gene. And glue, most of this RNA stays in the cell, and what goes into the EVs is a compilation of these three. Then you pick things like ASP, which only has one gene expressed. They're obviously both the same in the cell and in the EVs. And you pick something like alanine, in which almost this one gene is responsible for 75% of all of the RNA being made, and it winds up in the EVs. Valine, there's another outstanding gene that's selected for the expression. Proline, phenylalanine, lysine. And all of these indicate that there are very specific members of each of these families that are designated to be exported into the EVs. However, if you do the analysis according to the anticodon, all right, for each of these amino acids, they're virtually the same. So the bias isn't based on the anticodon, it's based on the sequence in the gene family. So whatever's recognizing that particular RNA to be exported out does not include the information in the anticodon. All right? Remember, the cleavage is actually occurring in the anticodon. One more thing that I neglected to mention is that these 5' and 3' ends, this 3' end contains the CCA, which is the non-templated trinucleotide that's put at the terminal end of the tRNA, right? So the processing of that 3' end happens after that modification occurs. So it's not happening while the tRNA is being processed and released into the cell. Okay, if you look at this cell line, BJ, and what I'm about to tell you is true for all of the cell lines we've now looked at. We've looked at 10 different cell lines and asked for the tRNAs, what is the most abundant tRNA 5' end we find in the EVs? And what we see by far, right, is valine. The valine 5' tRNA constitutes 62% of all of the tRNA sequences present in the EVs of 10 cell lines. All right. Arginine, that 5' end is 24%, and these others have roughly 10% or 11% contribution overall. And this is all the other tRNAs that we find in the EVs. So this brings with it a certain set of implications. It means that there is a mechanism inside of the cell that allows for the capture of a very specific gene, RNA transcript from a very specific gene family, right? In comparison to several other gene members of the same family. So even though the sequences look very much similar, there's something that recognizes some specific sequences. Why do I say it's a sequence? Because the structures, as you can well imagine, for all of these tRNAs are very similar. This cloverleaf structure is the same. So it cannot be that it's recognizing something different about the structure. And there are two possibilities. One, that there is a sequence recognition, whatever distinguishes one family member from another. And the other, which is even more, if I think, interesting, is that there is a modification pattern that's different on each of the family members that is being recognized. And we know very well that these tRNAs, these structures, have very specific anticodon and D and T loop modifications that occur for these particular elements. So in the case where we have this 5' end present, any modification from roughly 35 nucleotides on up to 1 could be a source of a recognition complex that is seen by the UVs. Okay. So there's a variety of things. Is that clear? Are there any questions on that? No?
So is this kind of a distribution specific to the PG base, or it's in general?
No, we've done this. This is true for 10 different cell lines. 10 different cell lines. Not only for PG. Yeah.
I think this is very, very interesting for that. That means that exome or microvascular, that some part of this RNA, they just biogenesis in the cell, but their function is not in the cell. They just send this out to the exome, and see this microRNA, this is not microRNA, this is a tRNA or something. The function is just want to affect some other cell. That means this may be a very, very important cell communication.
Right.
I don't know that this ARG or valuable. So there's specific this tRNA have function to affect other cell. Can you imagine why this should go out, not stay in the cell?
So we do not have an answer to that. We will hopefully have an answer pretty soon. I should tell you that most of what I've given you here has not been published yet, but it's in press. It's been reviewed and it's going to come out soon. So everything I talked about in RNY5 came out in an RNA paper in December of last year. And what I'm doing now is going to come out fairly soon. So all of this story is going to come out. The question that you raise is what phenotype might these RNAs, these 5' end fragments, have? And we don't know, but what we are very interested in testing is whether they affect translation, right? Whether they're part of either a repressive, negative repressor, or positive, or both, right? Regulatory element in the recipient cell. Now, it is not certain that that has to be the case, but we do know that that RNAs that an overproduction of a particular tRNA, right, has a negative effect on the translation of machinery in general. So it may be that this provides some kind of either impetus for improved translation efficiency or regulation. That's where we're looking right now. The other feature is that the same activity that cleaves RNY5 cleaves this molecule. All right. So, and it's only found in the EVs. All right. So we don't see the activity in the whole cell. We only see it in the EVs. So perhaps I want to retract that statement. I want to say that it is possible that it's the same because we don't have the candidate enzyme yet. What we know is that the same kind of cleavage happens and present in the EVs for RNY5 and for the tRNAs that we see.
Do you think that is an enzyme, that's protein, some protein, or maybe some RNA molecule? They just cleave this.
So we have tested that in both ways. We have denatured and destroyed the protein contents of the cells all right, of the extracts of the EVs, all right, and the extract, that processing activity goes away. The RNA is still intact from the cell. So if it's a catalytic RNA, all right, it may need a protein, but it's not only a protein. We've looked at the activity of the in RNY5 of the other RNA members that are isolated from it, and we do not see that actually that cleavage event happening using RNA as the potential catalyst for doing this. So we think it's a protein. It may be a complex of proteins, and we've been working. I have a postdoc who's working on this exclusively now for a while, and we have a couple of interesting candidates, but they're not known as RNases. So it would be kind of an interesting new activity if that were the case. But we definitely do not find angiogenin, which is known to be the cleavage in the cell for tRNAs, and it's not dicer, and so it's another activity. Okay, so in the last couple of minutes, I just want to sort of wrap everything up that we were just talking about. The way to think about this now is that cells are basically surrounded by literally millions, if not billions, of these molecules, of these vesicles. They are constantly being flooded and taking this material in and out. Why doesn't all of this affect all the cells all the time? Why isn't this chaos? Why isn't this a recipe for chaos? I think the answer sits right here in this room. The answer is that this room is flooded with a whole variety of electromagnetic waves. We have Wi-Fi, we have radio, we have television, we have a whole variety of electromagnetic waves flooding this room. All right? We're totally innocuous to it because we have nothing to respond that is capable of responding to it unless we have one of those computers or phones, right? That's the trick. It is the capability of interpreting the signal, not necessarily of restricting the signal, right? And so there is clearly what's happening is that there's clearly a sense of a need to generate plenty of signal, and evolution has apparently gone in the direction of interpreting the signal rather than restricting the signal or making it much rarer. The second thing that is very clear is that these vesicles are little factories. They're processing factories, right? At least from the two examples that we have right now, the yRNA and the tRNA stories. They process RNA explicitly in this compartment to be exported. All right? But remember, when I showed the pictures of the vesicles going into the cells, the entire vesicle goes in. It doesn't meld to the membrane, dump its contents out. The whole vesicle enters the cell and forms this perinuclear complex around the nucleus. That means that the same activities that we use to test to see what's cleaving these things is in these vesicles. And what does that mean? That means that these vesicles can act as amplification devices. They could take native substrates in the host cell and do the same cleavage. Right? That potential exists in this as a way to amplify the signal. We calculate that a given cell in a given in vitro, in a very specific environment, namely in the culture plate, all right, that we see from 1 million cells about 10 to 100 million vesicles. All right? So in other words, something in the order of about 10 to 1,000 vesicles per cell. That's not a lot of signal, right? Considering it's, they're diluted out in the environment that they're in. So if it's going to have an effect, if, for example, the cell death phenotype we see is going to have an effect, right, and the signal is not very pronounced, right, there seems to be either an incredibly active molecule, and the cell is very sensitive to it, or there has to be an amplification of that signal once it reaches the cell. Both of those possibilities are very interesting and clearly subject for additional studies. This slide is meant to summarize that. Basically, that the donor cells can determine the sequence of specific RNAs to be imported into EVs. How we know how this happens, we have no idea. We have not a clue how this happens. Number two, the processing activity of the EVs remains active in the recipient cell. That possibility is very astounding because it would represent a biological amplification system. And finally, the specific RNA processing occurs within EVs. Again, showing a very novel kind of biological strategy, namely, put a process into a vesicle whose purpose is to send outside and communicate with other cell types. So we think there is a large number of questions that need to be answered and opportunities for people to work on. And they include what are the inhibitors found in EVs that prevent such EVs from killing other normal cells. Remember, the phenomenon we saw was that if we take EVs from normal cells and treat other normal cells, nothing happens. All right? It's only when we get it from either the RNA out of the EV or synthesize the RNA itself. So there must be an inhibitor that's present in normal EVs that prevents the same fragment, the 31 nucleotide fragment, from killing other cells. Right? And if that is true, you can imagine a variety of approaches that could utilize that to minimize cancer enrichment. The enrichment factors. What are the factors selective capture of RNY5 and tRNAs? And we have, I never told you this, but there are other RNAs that look like they're being processed specifically in the EVs beside these two. Nucleases. What is the processing element that cleaves the tRNA and the RNY5? And as was asked previously, what are the factors that the fragment of RNY5 interact with to cause cell death? In triggering the pathway, the apoptotic pathways that we talked about. So these are incredibly, I think, interesting and scientifically important questions, and they're very different than the medical questions that people are asking for EVs today, where most of these are questions that have to do with biomarkers or delivery systems for drugs and things like that. So I think there is a path to sort of doing basic research here that is going to yield very novel and very interesting and important areas for our understanding of biology and medicine. So I want to acknowledge the people who did the work on this. This is Sudhito Chakravarti. This is Ashwin Prakash, who's done a lot of the analysis that I presented on in the EV stuff. And this is the brave postdoc who's been working on the processing enzymes for RNY5 and the tRNAs. So I think I'm going to stop here and take any questions or comments that you have. I'd really love to hear your opinions about what I presented. As I said, some of this is unpublished, and some of the ideas we talked about in terms of the biology that we did. So I'll leave the floor open and take any questions that you have. Thank you.
Thank you. Can you give us an example about such as a disease caused by or related to EVs, not just killing other normal cells?
Sure. So why does a cancer cell have lost? Why has a cancer cell lost this inhibition factor that is present in normal? Right? So there are two implications here. One is that the same RNA molecule that's capable as a synthetic molecule or in a cancer cell EV is capable of killing primarily primary cells. Okay? What inhibitor is lost into doing that? And what is the implication? One is that why does the cancer cell gain anything by losing the inhibitor? Well, what it clearly happens is that when you co-culture cancer cells with primary cells, you begin to clear the environment of any competing primary cell that's sensitive to the activities of these molecules. So it gives rise to in a competitive advantage sense in a cancer environment, right? To basically gather resources for the growth and spread of the cancer. Right? This is something that we're beginning to see in xenograph experiments, where we see the cells that with the xenograph die, and we see the RNY5 molecule in the mouse cells themselves, which must mean they had come from the human xenograft that are present. The other thing is quite striking is the fact that the RNAs themselves that are present are present at fairly low amounts, which means that these RNAs are likely to be found in these vesicles at very low amounts, but capable of being transported all over the whole individual in which the tumor might be present. And one thing that seems to be identified is that by a mechanism we don't understand, is that there seems to be accumulation of these EVs in distal sites which correspond to the sites of metastatic growth. So there's some preference, some localization of these EVs through the circulatory system to go for, like breast, to go to bone regions, right? And in doing so, there may be some kind of homing mechanism for when the release of cancer cells go that actually use these kinds of markers. This is all speculation. We don't know anything about the fact that this is truly available, but we do know that these pieces of evidence require sort of the controlled genetic experiments we can do now. So that's about the best I can give you right now, is in the cancer field.
Another question?
So a further question to my previous question. So you mentioned the distribution of the tRNA in the EVs remains the same in among 10 different sort of cells, right? So I presume if these kind of cells want to impact its neighbors, they impact differently because they are different cells. But why is the EVs, they remain the same tRNA distribution among all of them?
Yeah. So the answer to that, again, is speculative. But you can imagine that what's going on is that, as with the RNY5, the phenotype it wants to actually trigger is a phenotype that's operating in every cell. So if, for example, that the tRNA fragments have an effect on translation, right, you can imagine that what's being done by the EVs is bringing multiple elements, this being just one of them, all right, and where you want to control the translational functions in your recipient cell because you have other elements that are benefit from that kind of interaction. So the sort of universality of what we see for the tRNAs may be a strategy to be able to help whatever else is brought with it. Right? The cool thing about the EVs is that it's a multi-pronged approach. It's not carrying one thing. It's carrying many things. It carries microRNAs, it carries proteins, it carries these other processed RNAs. So it is an opportunity for a donor cell to have multiple kinds of effects by packaging all of these things together, right? And that's why I think the thought of an amplification system is necessary, because it's clear that while the contents of the cells are different, right, the proportions within the cells are likely to be also different. So, I think there is a need to amplify some things and tone down some other things, and that probably happens within the recipient cell.
So back to the case of the Wi-Fi. Is the Wi-Fi from primary cell different from the Wi-Fi from cancer cell?
No.
Totally the same.
They're totally the same. Exactly the same cleavage site.
Okay.
Yeah. And pretty much the same amounts also.
Okay. So my second question is, what happened to the students you mentioned who's initiates this project? Is he still working on this one?
No,
the
he's
brave
gone.
one.
Yeah, the brave one is gone. He's now, he decided to go work for an exosome company. So he's working for a company in Boston called Exosome Inc.
Still same area.
Yeah, the same
Okay.
area. Yeah.
Oh, nice. Thank you.
Question?
I have a question that, so if my understanding is right, so across the 10 different cell lines, the EVs all have the same portion of the valine TRAs. They all 62% of valine TRAs in these 10 different cell lines, right?
Mm-hmm.
Okay.
The
So,
fragment, 62% of all the tRNA that we see in the EVs come from the 5' end of valine, from the one gene.
that's good. So it means that maybe it is not just the the TRA is not packaged into the EVs just by accident.
Right.
Maybe it is regulated. So the absolute number of the TRAs packaged into EVs, is that significant, large enough?
Yes, so it's one of the more abundant RNAs that are in terms of mass. tRNAs and microRNAs are the two largest groups of biotypes in the EV itself.
Yeah. So have you checked are these TRAs packaged into the EVs with this target amino acid, or is just the TRAs?
With its target, I'm not sure.
With this amino acid targets, I mean the valine TRAs with valine.
Oh no, there is no. These are processed before the anticodon interacts with the amino acid. So we don't see any evidence of coupling of the histidine, for example, with the histidine.
Okay, cool. And the next question: have you seen the valine or arginine-rich mRNAs? Okay, I will talk to you later.
I think your results is very interesting. So, what do you think that this EV, this RNA, I mean, you know that this can kill the normal cell death. We know that cell, we have the contact lead to inhibition or lead to death. So I wonder that this EV, this RNA in this EV, does that mean that working locally? Because of normal cell, usually when they contact, they just inhibit, but human cell not. So does this mean just because this RNA in EV that works locally, so to come, but the tumor cell, they have resistant to this contact. So that I think that this RNA may be working locally or far away. Locally maybe easily or something.
Yeah. We don't. It seems from the titrations that we carry out that you need a fairly large number, or the titer of the RNA needs to be relatively high, right, in order to see an effect. So the effect of of the EVs is probably most pronounced in the local environment. All right, but again, if we are correct, then the release of these EVs into distal environments bring with it the opportunity to make more fragments once it enters into another cell. All right. Now, whether that happens or not, we don't know. But it is an intriguing idea that if there is some homing device for the EVs to get to a particular cell type somewhere else, you can see this as a means to accumulate the EVs in this recipient cells and generate more of this RNA. All right, but that's all speculation.
Okay. So another, we know that the exome, this RNA, the 5 and they have 3 phosphors, something. It's just like a virus. I read some papers that in cell, they say that RNA in this EV is different from the cell. It's because this EV cell, we know that the virus RNA and our cell RNA is different. The 5 and they have 3 phosphors or 2 phosphors, something, I forget that.
Yeah.
But it's different. So does that, in this case, this EV kill that cell? It's just like the virus infected the primary cell, so makes this cell dies,
Yeah.
or something.
So just to bring this into context, Pol 3, when it polymerizes, those transcripts are not capped.
Mm-hmm.
There's a triphosphate at the 3' end, an uncapped triphosphate.
Mm-hmm.
The question and many viruses have this also this triphosphate at the
Yes.
5' ends. So one possibility is that these 5' ends of the RNY5 that we see, require this triphosphate to function. We made in the synthetic version of the 31-mer a monophosphate version, and that is active.
Still makes
Yes,
sense.
right.
Okay. So this I will count maybe in Chinese.
这个就是Tom 今天上午的报告，我觉得还是非常非常精彩的。对这个 exome 研究，我觉得就是说，我们现在比如说大概去年前年有些 cell 文章，我们国内现在跟风似的，一下子就在搞这种东西，就是说外泌体啊什么怎么去做，然后就用细胞有没有什么做，就像反应作用。我至少知道，就是说很多人在做这个东西，但是你可以发现 Tom 的研究完全是跟我们不太一样，对吧？他完全还是从刚刚讲非常 basic 方面的东西。所以我觉得，就是从研究角度来讲的话，是那种研究可以分子高、分子低，对吧？但那种跟风式的这种研究，这种去做的话呢，你肯定也得不到什么结果。因为我们实验室其实5年前我们就在做 YBT，5年前我们就在做这件事情，并不是我们现在才干。我们觉得现在我们这边上现在还没有发，所以对 YBT 这一块研究，就是说你从这种学生那天说，于老师看见我看到 IPS 好多人在做 YBT，我们怎么办？我说你不用管它，对吧？你做的东西它不一样。所以我就说外媒体研究这一块，像Tom 今天给你提出了非常多的问题，所以尤其是他好几张 slide， 所以我觉得就是你只有做这种访问的问题，然后你才能把外媒体研究清楚，然后才移动。如果我们还是把外媒体提出来，我们现在大家都问，我看训练提怎么提啊？然后加入细胞有什么作用啊？都在干这种事情，这件事情，这种只是一个工具，然后你要研究非常重要的问题，然后对这个东西去解读，我觉得可能会非常非常重要。而他今天提的很多问题也是非常 unexpected， 包括刚才我说那5.0，他原来那篇 Cell 文章说为什么会有症状，他说因为是有5.0，而这个0呢会跟病毒要去识别，而他的结果完全不一定是这个，说明它是一种 specific 这个从 RNA 里边在起作用。所以我说从研究角度来讲的话呢，这一块可能还是非常值得大家去思考。包括 YMT 这块研究，我们实验室我大概在2-3年前那篇 Cell 文章发，我在科学中报当我们约这个 RNA， 我们当时的5张题目就是 Moving RNA Forward， 当时题目就这样，当时就提到了这些 communication， 可能它就是它从哪来的，它中间怎么去作用，怎么去进，怎么去弄。但是那篇文章很可惜引的不是很多，但这2天最近好像引的现在越来越多，就是对这个 Moving RNA 整个去理解。所以我在讲，就是说，从我们做研究角度讲，你能不能在5年前，或者说尤其我们现在，你5年前你就觉得哪个地方可能会非常做，你就开始做，而不是现在才变好的，然后你再踏入进去，再跳到这个坑里边，那估计就上不来了。 对吧？所以对研究者Tom今天他其实给了你一个非常好的 base， 从哪儿着手，对吧？外媒体这一块，大家现在国内外媒体非常热，你能够从这些很基本的问题去着手，然后去做研究，我相信你可能会有非常多的这种不一样的这种结果，对吧？然后我们热烈掌声对Tom今天的表现感谢。
So can I just make one more comment? I want to go back to the very first day in the morning when I said that if anybody has additional questions that occur to you after the meeting, please feel free to contact me. And if anybody in the audience is sort of interested in the work that I've presented in both the long non-coding RNA or the EVs, then please contact me if you have interest in working in this area too. Thank you very much.